have MHC class 2. Now, what's a professional antigen-presenting cell?
A dendritic cell with
a dendritic
antigen.
cell.
With the antigen presented on the cell.
Another really good example is a B cell. Is a professional antigen-presenting cell. So,
Why are they called professional?
because they do it for a living. Now, there are other cells that can be induced to express MHC class 2, and those would not be called professional. But MHC class 2, professional, they do it for a living, that's their job. Dendritic cell. So a dendritic cell presents antigen, right?
Yes.
Presents antigen on the MHC class 2 molecule to what
T
type
cell.
of cell? A T cell. Okay, be more specific.
The CD4
Helper?
Yeah, CD4
She's going over
or
this.
helper T
T helper
cell
cell. Yeah.
presents antigen, right? A B cell expresses MHC class 2.
It doesn't have an APC.
It is a professional. A B cell is a professional APC. Does that surprise you? Does that just blow your socks off?
Because he talked about it yesterday. Because the B cell and the T cell needs to activate in order in the germ level to present to have the specific antibody.
Exactly. In order for B cells to respond to protein antigens, they have to induce a T cell to help it. A CD4 helper T cell has to help B cells in order to get them to respond to a protein antigen. So it has to show or present the antigen to the CD4 cell. So the B cell binds to antigen by its antigen receptor, which are, what's the nature of the antigen receptor on the B cell? It's an immunoglobulin, right?
Yeah.
Yeah, it's a membrane-bound immunoglobulin. So it grabs onto this antigen to which it's specific, and then it internalizes that antigen. Okay, brings the B cell receptor and the antigen get dragged inside the B cell. And then the B cell chops up the antigen, processes it, right? And then in endosomes, right? It's within processing it in an endosome. And then in another endosome that contains the MHC class 2 molecule that was just made. Remember, this MHC class 2 is a membrane molecule that's being made all the time. Doesn't matter what else the B cell is doing, it's making the membrane molecules. So it's a membrane protein, so it gets made, on the ribosome, gets into the Golgi apparatus and goes bloop, 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 up to the surface, like any good membrane molecule is going to do. Okay? So the antigen gets united with the MHC class 2 in one of those vesicles, sticks itself into that peptide binding groove, is everyone with me? Goes up to the membrane and is displayed there for a T cell with antigen specificity for that antigen. So you remember yesterday how Dr. Gregg talked about that, right? So a B cell and a dendritic cell are the cells that express MHC class 2. And that MHC class 2 is the molecule that presents the antigen to CD4 helper T cells. Now, do you know, if I asked you to draw that, which I ask you to do in this packet here, if this is the B cell and this is the T cell, tell me what to draw on this B cell. It's presenting the antigen.
A receptor?
So I'm going to draw the what?
BCR, right? Or are you
Well,
asking for?
okay.
Are you asking for?
I draw the B cell receptor here,
Yeah.
right? It's bound to the antigen. It gets endocytosed,
Is it this one?
chops it up.
She's doing a different one. She's not talking about a T cell. She's talking about B right now. Yeah, the CD40.
These two vesicles merge, and we've got this. What's this thing? That's the CD40.
to the CD40, CD40.
What did I just draw there?
Ligand, what is it, toll receptor? No.
That's the FC receptor.
This
No,
would be,
that'd be
in
an
order for
antibody.
this B cell to present antigen to the T cell, this has to be an MHC class 2 molecule. Right?
Right.
All right. Now, what's that up there?
That's the
Antibody.
Immunoglobulin.
That's it's B cell receptor. That's the BCR, right?
Yes.
So the first step is that the BCR binds to the antigen. The next step is that the BCR and the antigen are internalized. The antigen is chopped up into little bits inside this endosome. Here's another endosome with an MHC class 1 molecule. Class 2, sorry, class 2. Don't make that mistake.
It might be an emergency.
No. It's just my husband saying hello.
Oh, okay. That's sweet.
So, anyway, so one of the peptides that was chopped up here gets into that peptide binding group right there and is presented now on the membrane of the cell, of the B cell. Now, what about the T cell? What do I need to draw here?
T cell?
A dendritic cell.
The T cell, here's a T helper cell.
Mm-hmm. T.
So
TCR,
what
right?
molecule am I going to draw here?
TCR.
A TCR. Okay, so here's the alpha, here's the beta chain of the TCR. Okay? All right. What else do I need to draw?
You need to draw a 40/40, the signal 2, right?
A what?
40/40 ligand. A co-stimulation. You need
Yeah,
co-stimulation?
but before we co-stimulate,
Mhm.
what's going to hold on to that MHC class 2 from the T cell?
The toll like
CD what?
4.
Thank you. CD4. All right. And then we also have co-stimulatory molecules. So I got to move that CD4 over a little bit. The label. But that's the CD4 molecule right there, right?
Mm-hmm.
All right. Then we have co-stimulatory molecules.
My arm's gonna fall out.
And what are these?
Co-stimulatory.
Well, they're co-stimulatory molecules, but give me some names.
Immature.
CD40 and 40 ligand.
CD40 and
40.
CD40 ligand. Do we have
Or toll-like receptor.
CD8086 and CD2?
No, we know it's 40/40.
Or you just learned the 40/40 ligand? Okay.
Mhm.
40/40. CD's 40/40.
Yeah, okay. All right, so that's the interaction of the B cell and the T cell receptor and CD4 with the MHC class 2 and so on, okay? Yeah,
I just wanted to make sure that second circle in the B cell is the MHC, right? That one.
which one?
That one.
This one here contains it contains the MHC class 2 molecule. Because when you make a membrane protein, any cell has to make membrane proteins, right? And they get made and then they get shuttled into the Golgi, which has these little vesicles that bud off from the Golgi, right? So that's what that is. It's already made.
MHC2
it's gone through the
molecules.
endoplasmic reticulum into the Golgi. It's bud off, and now the little endosome, the little bleb, is going to merge with this one here. So I don't have that drawn very well, but it'll merge together like that. And then, okay, so those are all bits and pieces of that antigen. And then one of those pieces of antigen is going to go into that peptide binding group of the MHC class 2 and go to the membrane there and be expressed on the membrane. And you have some really good pictures of that process in your book. And the same thing would happen if I changed this from a B cell to